So I'm really excited to host today Dr. Robert Paul Juster. I always love listening to his talks because I learn so much. So without further ado, first let me do a land acknowledgement once I can advance my slide. I want to recognize that I know we're all coming from very different countries, but I want to recognize that we at the cluster, many of us live, work, and play and participate in a community on the unceded, ceded, and traditional territories of the 203 First Nations, along with the 38 Métis Charter communities, each of which possess their own unique traditions and history here on this land that we now refer to as British Columbia and Canada. We acknowledge the importance of the Truth and Reconciliation Commission of Canada's call to action, the United Nations Declaration on the Rights of Indigenous Peoples Act, and the BC Declaration of Rights of Indigenous Peoples Act. In all of our work, we are committed to ensuring Indigenous women's rights to health and safety and equal opportunity to participate in a manner that recognizes and respects Indigenous cultures and traditions. And I really urge any of you that are not in Canada to acknowledge and learn more about the Indigenous communities and lands on which you dwell. Today, I'm joining you from North Vancouver, which is part of the unceded and traditional and ancestral homelands of the Coast Salish peoples, the Musqueam, Squamish, and Tsleil-Waututh First Nations. These talks are brought by the Women's Health Cluster. We have over 400 members, actually. That's an old slide. A number of events, also an old slide. It's got some 20, but one thing that is going on right now is a brain health conference on hormonal contraceptives, so check that out. We run a number of trainee awards, so any of you trainees that are out there, we have some awards for attending the brain health conference, but we also, and as I think I said this already on the hormonal contraceptives on the brain, but we have some other ones as well for travel awards. So please take a look out for those and become a member, support us if you so desire. So I'm going to stop sharing my screen so that you can hear from the person you really want to hear from. You are really in for a treat because Rob always gives a fantastic talk. And let me just read out his bio to you. He's the director of the sex asterisk gender, we'll have to ask him what that means, Allostasis and Resilience, based at the Research Center of Montreal Mental Health University Institute. Dr. Juster is an assistant research professor in the Department of Psychiatry and Addiction at the University of Montreal. Dr. Juster also holds a CIHR Sex and Gender Science Chair. His research focuses on stress and resilience using a sex and gender lens, and he has dedicated his career to advancing, you're going to make me tear up, the allostatic load model first developed by the late and great Bruce McEwen. I just put his photo in a talk I'm giving in a couple of days. As many of you know, I like to read out people's fun fact, and Rob has a really great fun fact for us today. He dreams of one day of voice acting as he imitates colleagues, and I don't know if he imitates me, and great movie characters like Hannibal Lecter, Dr. Evil, I want to hear that one, General Patton, and many, many more. So with that, please take it away, Dr. Juster.
Thanks, Lisa, and thank you for the invitation. Today's presentation, I'm really very glad to be able to give you a real rundown of a lot of my work in more than 15 minutes. I think many of you have seen permutations of this presentation crammed into 15 minutes, so it's a real treat to be able to give it to you in its entirety. Can you all see my screen? Great. I'm just having a hard time moving. All right. So we know that there's important sex differences between men and women, especially when it comes to mental health. This is pretty much one of the main reasons why we're interested in studying sex differences. However, I'd like to point our attention to the fact that a lot of these conditions can change over time. So cardiovascular disease used to be something that killed far more men in Canada than women. But there are now more women in Canada dying of heart disease every year than men. What's happened? There's definitely been changes since World War II in terms of representation in the workplace, as well as women have not benefited from as much support in the household, even though greater participation in the workforce. We know that there's important biological differences in terms of a lot of the risk factors and the treatments of cardiovascular disease. However, the real central point is beyond biology, we really need to consider sociocultural gender when looking at a lot of these stress-related conditions. So I think I don't need to tell this group what sex and gender is, but I will anyway, just for the purposes of the recording. Sex really refers to all the biological attributes of humans and animals, including physical features, chromosomes, gene expression, hormones, and anatomy. Very independent from sex, but often similar in terms of construct development is gender. And this refers to all the sociocultural roles, behaviors, expressions, and identities of girls, women, boys, men, gender, and gender diverse people. So I liked taking the slogan that was first developed by the Institute of Gender and Health: every cell is sexed, every person is gendered, and every organism is stressed. And what do we mean when we talk about stress? Stress itself is a very complex term. A lot of people feel that it overlaps with all kinds of things like anxiety, fear, pain. So the definition I'm going to use is one really central to Bruce McEwen's work, which is that stress is broadly defined as a real or interpreted threat to an individual's physiological and psychological integrity that results in biological and behavioral responses. There really are all kinds of traditions that can be used to measure stress. So an environmental perspective will ask people about objective stressors. So did you grow up in a poor neighborhood? Are you exposed to discrimination? Related to this is the person's subjective experience of this objective stressor. So this comes into the psychological domain of subjective distress. So on a scale of 1 to 5, we'll ask people how stressed they are. And then finally, a biological perspective where we will look at biomarkers of stress. So the stress hormone cortisol, blood pressure, immune functioning. Many of these traditions are studied kind of in isolation and in disparate disciplinary silos, but I'd like to argue that it's really central to try to integrate them as much as possible in the research that we're doing with humans. I don't think it's as easy to get a subjective distress scale from a rodent. So when we're talking about the biological stress response, we're really talking about two main axes. So in blue is the sympathetic adrenal medullary axis release of adrenaline, noradrenaline, and dopamine. And in green, several minutes later, is the hypothalamic pituitary adrenal axis release of cortisol. I've spent the better part of my career stressing people out and collecting their spit. We can measure saliva we can measure cortisol very easily in saliva. Oops, I'm sorry. And so what we do is we bring people into the lab and we stress them out. And you'll see right off the bat here that men actually, in general, show a higher cortisol response to stressors that involve challenge. So the stressor that we use the most often is the TRIAR social stress test that involves 5 minutes of mental arithmetic, 5 minutes of a public speech in which you're sort of pretending to apply for a job, and then 5 minutes of mental arithmetic. And this task seems to be more stressful for men in general. We assume birth assigned men, but up until recently, people were not looking at gender identity in this literature. I will say that there is some research showing that social rejection as a paradigm seems to be more stressful for women. This is some work by Laura Stroud at Brown University, where if you have a situation where participants are increasingly socially rejected by Confederates in the lab, that seems to be more salient for women. So I just want to emphasize that point, that the nature of the stressor could be potentially something that's related to sex and gender. We can also look at cortisol and stress hormones over the course of the day to look at day-to-day variation in cortisol. And in general, we don't tend to see important sex differences in diurnal cortisol in humans. Studying all this is really important because these profiles give a real portrait of potential risk for stress-related diseases. So what you see here in green is the normal response. We have what's called the cortisol awakening response when we wake up in the morning. It's adaptive, we want to have it. And then lowest levels just before going to bed. In red is the profile that you generally see in depression, so the hypersecretion of cortisol. While in blue, this is what we see more in burnout, but also conditions like post-traumatic stress, fibromyalgia, chronic fatigue. And this is important pharmacologically because take, for instance, a depressive patient that might come in for burnout or sick leave rather, but their physiology is completely different. You give them antidepressants, you might be lowering their levels of cortisol, which might not necessarily be a good thing for someone that has already low levels. All of these changes in terms of stress reactivity and stress responses and physiology are really well subsumed by the concept of allostasis. So allostasis, moving beyond homeostatic mechanisms, is really how the body and the brain adjusts to environmental demands. So the central principle of allostasis is that to maintain stability, an organism must vary all parameters of its internal milieu and match them appropriately to environmental demands. So the best example of this is an actual stress response. And this work was taken into the field of substance abuse, and the concept of allostatic states was developed. So here you have a normal response. Someone stresses out, they chill out once the stressor is gone or recover. This is what you want as a stress response. There's 4 profiles that have been proposed to start denoting some potential risks. So one is repeated activation or repeated hits. So you can think of someone who lives in a dangerous neighborhood. Related to this is a lack of adaptation. So people who are exposed to similar stressors who are unable to habituate. A prolonged response where the recovery takes longer to happen. This is quite problematic in the context of a lot of aging research. And finally, an inadequate response. So the example of burnout, where there's simply not enough production of stress hormones or whatever parameter you're looking at. So just to do a recap, allostasis at its core is adaptive, it's stability through change, but this can become an allostatic load or wear and tear on the body and brain over time. And this is really the work of Bruce McEwen and colleagues that really pit the brain at the center of all of these adaptations, something that sort of boring homeostatic theories couldn't really do a good job of. And Bruce really pitted the brain at the center of all of these different adaptations to propose the allostatic load model, which is, in my opinion, and I think many people's opinion, one of the most comprehensive models of chronic stress. So by definition, allostatic load is the wear and tear of chronic stress in synergy with unhealthy behaviors. You can think of it as sort of the straw that breaks the camel's back. And it's not just cortisol. We're looking also at metabolic biomarkers, immune functioning, cardiovascular functioning. A good layman's tool is to think of these 2 little kids on the seesaw going up and down won't really weigh the seesaw down or wear it out. But if you imagine 2 sumo wrestlers doing it out over time, the seesaw will wear. So allostatic load is essentially the seesawing of different biomarkers, but for an entire network. Another way to think of it is as a domino effect. So in theory, cortisol, catecholamines, and immune biomarkers become dysregulated. This leads to a lot of changes in cellular functioning, and that increases the risk of a lot of metabolic and cardiovascular biomarkers becoming dysregulated. And collectively, as a kind of domino effect that's just trailing down, this increases the risk of morbidity and mortality. There is a temporal disease cascade that has been proposed, as I mentioned. So first, it's really thought that neuroendocrine biomarkers are really central to causing this cascade. And some work by Martin Picard at Columbia has really emphasized mitochondrial and cellular dysfunction as central to this process that then leads to what's called the secondary and tertiary outcomes. So the metabolic and cardiovascular parameters that become dysregulated and increased risk of stress-related diseases. I have a paper with Martin that just got published I'm quite proud of that does a nice job of really articulating our point of view with regards to energy is really central to allostatic load. And I'm just going to use this diagram to recapitulate the distinction between allostasis, allostatic states, allostatic load, and allostatic overload. So allostasis, you'll recall, it's adaptive. We want to have these stress responses in order to adapt to the environment. But as time goes on and this becomes an allostatic load, these different systems become dysregulated collectively, and that increases the risk of allostatic overload, which is a concept that was brought up in evolutionary work by Wingfield that articulates that people who are producing too much energy or not enough will lead to a lot of diseases. And they use a lot of really interesting work with birds and migration to really hash out this idea. In humans, allostatic load can be measured by looking at a battery of different biomarkers. So this was the first studies that were done looking at successful agers. So these are older adults that are doing pretty well, but they thought, okay, well, let's try to find a way to predict those that will develop problems over time. So they took these 10 different biomarkers that are all in some way related to stress. And what they did is they didn't just look at clinical cutoffs, they looked at subclinical cutoffs. So for biomarkers where high levels were problematic, they used the 75th percentile of the distribution of that biomarker for their sample. And for biomarkers where low levels were problematic, so DHEAS or HDL cholesterol or good cholesterol, it was the 25th percentile. And this really became like the standard way of measuring allostatic load based on biomarkers and their distribution for your particular sample. Now, I'll just point out that most of the time this is done without any consideration of sex. And some of the work that I'm going to present today will show you the importance of actually not only considering potentially sex differences, but also gender diversity. One of my colleagues, Zachary Dubois, said, yeah, it's good that you consider sex, but maybe these biomarker distributions should also be considered according to gender identity. Another way that allostatic load can be measured is actually basing it on clinical reference ranges. So not necessarily waiting for a biomarker to pass a threshold, but using the 75th percentile for that particular range. I'll give you an example here. So here you have 7 because this person's cholesterol is high. They're given statins because it passes the 5.2 threshold. But this person actually developed diabetes in 2 years. If you look at their glucose, 5.4, it's not quite at 6, but it's almost there. Triglycerides 1.51, not quite 1.8, but almost there. HDL cholesterol is fine because high levels are generally considered good. So for these 4 different biomarkers, this individual has 3 of them that are passing the 75th percentile. So they would get an allostatic load score of 3. Most of the studies that have been done have used a minimum of 10 biomarkers, and most of my studies use 20 biomarkers. And what you see here on the x-axis, this is an old review, but the tendency still applies today. On the x-axis, you see the number of studies and the representation of these different biomarkers in these different studies. I'll just point 2 things out. There's a lot more interest nowadays in immune and inflammatory biomarkers, and also cardiovascular parameters in a more high frequency metric. So rather than just looking at resting heart rate or resting systolic blood pressure, people are incorporating measures of heart rate variability throughout the day or in the context of a stressor. So that's going to be really interesting moving forward, because I think sex differences are going to emerge when looking at different biomarkers that might be different between the sexes or by different gender, people with different gender expressions. Most of the studies were done in public health, and in a pretty consistent way, allostatic load predicts cognitive decline, cardiovascular disease, increased mortality. And the real advantage of the allostatic load model is it's not just looking at one biomarker at a time, but several. And it's not just looking at clinical reference ranges, it's looking at subclinical ranges. So it makes sense that it moves beyond something like metabolic syndrome or moves beyond the Framingham risk factors for cardiovascular disease. And what comes out from the epidemiology work is that increased age is by far the most standard covariate to use, and it's the most important predictor of increased allostatic load. So pretty much from the ages of 20 until 60, we have increasing allostatic load, and then it plateaus off from about age 60 to 90. Other really important causes of allostatic load are low social economic status, non-white race ethnicity, and in a very inconsistent way, sex differences. And that's really the next part of my presentation that centers on sex and gender in relation to allostatic load. This little diagram shows sex and reproductive events and sexuality as these little triangles over here. But you could imagine that sex and gender are also going to influence health behaviors. They're going to be influenced by genetics, and they're going to be influenced by a whole bunch of life experiences. So while my presentation really centers on these 2 little triangles here, I'd like you to sort of think about how it extends to potentially many different causes in direct and indirect ways. Allostatic load is usually indexed using metabolic, neuroendocrine, immune, and cardiovascular parameters. And in a pretty, across the board, allostatic load is predictive of neurological atrophy, mobility limitations, functional impairments, cognitive decline, psychiatric symptoms, cardiovascular disease, and all-cause mortality. That distinction between physical and mental health is something that I've really devoted my career to trying to contribute to, in terms of trying to show how allostatic load can be used to not only to predict physical health problems, but also psychiatric symptoms. And of course, I'm very interested in asking that same question, but with consideration of sex differences and gender diversity. And that was really the core of my career throughout my PhD and now as an assistant professor. Just to recap sex and gender, what we mean, this was a graphic I grabbed from the Lancet, and I just kind of thought it was interesting that attributes for females was considered fragile and emotional, while for males, it's attributes like risk-taking and aggression. So even publications like Lancet can need a little update from time to time. In the rest of my presentation, I'm really going to break down work that I've done and work by my colleagues. We're looking at birth-assigned sex, sex hormones, gender roles, occupational gender roles, sexual orientation, and gender identity. And I'll conclude with some current work that is going on in my lab. So first, with birth-assigned sex, I had the pleasure of publishing a paper with Lisa's journal a couple of years ago with one of my students, where we did a systematic review of the literature where there were actual sex differences in allostatic load. And what we concluded was pretty much, if the study, because there are some studies that didn't even consider sex, but in studies where there are sex differences, men tend to have higher allostatic load than women. However, when we actually start looking at some of the sex differences among sex differences among women, they're actually related more to sociocultural gender. So for instance, women that attend religious services more often had lower allostatic load than women that didn't. This wasn't a study, this wasn't a finding in men. Social networks were also quite gendered in regards to older adults. So older Taiwanese women did not benefit necessarily from having a husband, but benefited much more from their same-sex social networks. So a lot of these different causes or contributors to allostatic load were showing this sort of gendered tendency that I really wanted to focus on during my PhD by looking at beyond sex, looking at sex hormones, gender roles, and sexual orientation. So first, for sex hormones, I mentioned before that men in general seem to show higher cortisol reactivity than women. However, this is affected by oral contraception. So women that are taking the pill show lower levels of cortisol when being exposed to a stressor. There's effects of menstrual status. So women in the luteal phase show higher levels of cortisol. For diurnal cortisol, I mentioned there weren't really very many differences. There seems to be some difference if you look at people about 45 minutes after awakening. But overall, there isn't really any important sex differences in humans. Now, all of these studies have one critical limitation. They don't actually measure sex hormones. They assume some variation based on the status or use of a molecule, but they don't actually measure it. So I wanted to do that during my PhD in a sample of psychiatric hospital workers. There were 3 times more women. I just want to point that out. Men are very afraid of blood draws for some reason. And this was our protocol. It's going to repeat itself throughout this presentation. People come in for a visit where we expose them to the CIHR social stress test. We take lots of measures of saliva and cardiovascular functioning. People go home, they spit some more so we can look at diurnal cortisol. We look at sex hormones as well in the morning, sex hormones before the stressor. And then they come back for a second visit where we do a blood draw and then compensate them. So this is a really standard psychoneuroendocrine protocol, and it's pretty much, I still use these protocols in my lab today. This is the CIHR social stress test that I mentioned. So it's got an anticipatory phase and then 10 minutes of the stressor. We measure diurnal cortisol at 5 different time points over the course of 2 days. And we measured sex hormones before the stressor and as well in the morning. Now, here is the graph that we normally see in the literature. Here you see that men show higher levels of cortisol reactivity than women, to the TRIR social stress test, no differences in diurnal cortisol. But when we actually adjust for testosterone, estradiol, and progesterone in the sample, we lose this sex difference in reactivity, but we're able to unmask a diurnal difference where women are showing higher levels of cortisol in the morning compared to men. And this, interestingly, was driven by higher levels of testosterone in older women, as well as women that had kids at home. So there's that interface between sex and gender going on and potentially in this result. So the conclusions from this is that sex differences in cortisol functioning are eliminated or emerge when adjusting for sex hormones, and that these individual differences must be accounted for when endeavoring to tease apart gender-based effects. So all of the future papers or slides that I'm going to be presenting, we're actually adjusting or controlling for sex hormones in our analyses. I'd like to also think that sex hormones is a way of actually looking at sex continuously rather than just as a binary male or female. And this is some work again by Martin Picard that really tries to emphasize the importance of sex and gender perspectives in mitochondrial science. Now, turning to gender roles, one of the first studies I ever did in my career was looking at cognition and gender roles, which are masculine and feminine personality traits that are really outdated. We used a questionnaire called the BEM Sexual Inventory, which was developed in the 70s. However, to this day, men tend to show higher levels of masculinity and women show higher levels of femininity for a lot of these traits. So 10 years ago, when I started my master's, I threw this into one of our studies. And my thinking was that gender roles are the social norms we assimilate and endorse via the enactment of sex stereotypes. And there's a rich literature that was done in Scandinavia that found that women in non-traditional occupations, so engineers, managers in the 80s, experienced greater distress, espoused greater masculine gender roles, but also had more stress-related problems, so higher levels of neuroadrenaline, adrenaline, and cortisol. And so I was interested in seeing whether gender roles would be related to allostatic load. And so I had the chance of just doing a small pilot project. And here, the more negative the number, the more masculine the person's gender role, and the more positive the number, the more feminine. And so we saw that higher allostatic load was related to greater masculinity. If you were a woman with greater allostatic load, if you were a woman with greater masculinity, there were also more physical symptoms. And finally, physical symptoms were correlated with allostatic load. So we used this as preliminary data for a CIHR grant, but then funded the rest of my PhD work. It was really the first study to look at gender roles in relation to allostatic load. With my large sample of 200 workers, I wanted to go one step further to test one of BEM's central hypotheses. So BEM proposed in the 70s that people who espoused high levels of masculinity and high levels of femininity would be people that she considered androgynous. So people that were able to adjust to the demands of the situation without acting in a rigid sort of sex-typed way. By reverse, those that were considered undifferentiated were those scoring lower on masculinity and femininity. And when I say low or high, this was based on the median splits that are being done in the sample. So not the fanciest statistics, but a nice way to sort of parcel out your sample into 4 different groups. So masculine typed, feminine typed, androgynous, and then differentiated. And so I wanted to test this out very simply with allostatic load. First, for mental health, we found, consistent with BEM's theory, that those with more androgyny showed higher self-esteem, more well-being, and less depressive symptoms. Now, for allostatic load, we measured it in 2 different ways. So one was the way it's normally done, what I'm calling all-inclusive, where there was no consideration of sex differences in the biomarkers. And then the second was with a sex-specific formulation. So using cutoffs that were specific to men and specific to women. For the allostatic load index that didn't consider sex differences, we found what's consistent in the literature. Men have higher allostatic load than women. But when we use the sex-specific formulation, we unmasked a sex by gender role interaction, where androgynous men showed lowest allostatic load compared to undifferentiated men, and undifferentiated men showed the highest allostatic load compared to undifferentiated women. I'll point out there's no differences among the women, and that was something that sort of vexed me, because we had 3 times more women, and Bem's theory, we hoped would apply to androgynous women, but it didn't. So just in terms of conclusions, so partial biopsychosocial support for Bem's theory, but it really begged the questions, why is androgyny not adaptive for women? Well, we thought about it, and it's like, well, androgyny might not necessarily incur the same benefits to women that have to do a lot of household responsibilities. This could be related to gender inequalities, multiple roles of women. This could be cohort effects. So this is something we were going to be studying further when looking at occupational gender roles. So we said, all right, we have people's individual gender roles, but why don't we look at their occupational gender roles? So we did a survey where this is just to show 1960s stereotypes of different professions. We reached out to people with 80 different professions in our sample, and we asked them to use the same scale as the BEM sexual inventory to rate what they felt was the masculinity and femininity of that profession. And what we did is a structural equation model to see how it would relate to mental health and to allostatic load. For individual biomarkers, we had a lot of associations predictive of mental health and allostatic load, but when looking at occupational gender roles, these were only related to workplace stress. So effort reward imbalance, psychological demands in the workplace, and decision attitude in the workplace. So we want to explore this a little bit further in other professions than people working in the psychiatric hospital. This really taps kind of indirectly at the concept of gender relations. So how men, women, and gender diverse people are sort of expected to behave based on their different professions. And even more macro than this is moving towards more institutionalized gender. The different policies put in place that can affect the health of people. One example of this is looking at occupational status. So this is based on people's professions and creating a rank based on census data. And this was a kind of a spurious finding with Bianca D'Antono, where we found that surprisingly, men with higher occupational status actually had higher allostatic load, but we saw completely the reverse in women. And when we started scratching the data a little bit more, we were finding a lot of associations with job control among women that was very different than what we were seeing among men, especially younger men. So I think when we're looking at these sex and gender differences, it's also really important to consider workplace stress as well as age. This is just pointing out that there's many different ways that we can index gender even more macro than the individual. I think a lot of Louise Pilat's work looking at gender indexes, as well as Greta Bauer's work with intersectionality is really important to consider moving forward in this literature. This is a beautiful graph that was part of Meet the Methods with Greta Bauer, and I really encourage you to look it up if you haven't already. I sort of am repeating myself now, but once again, it's really important to look at gender norms and institutionalized gender. And I think this is extremely important in the context of what's going on in the US with reproductive rights among women. And I think there are really interesting ways to try to look at where people are living and what are the laws in place that protect men, women, and gender diverse people. So the last 2 parts of my presentation are going to focus on sexual orientation and gender identity. We know that lesbian, gay, bisexual, and transgender people experience more stress-related diseases. And this is thought to come from sources of distal stress, so discrimination, victimization, structural stigma, so where people are living and whether they have laws that protect them, as well as proximal stress processes, so internalized homonegativity, and where people are in different processes of their sexual or gender identity formation. There was a really important report that came out about 10 years ago that really put a priority on LGBT health for the Institute of Medicine. And this largely was based on a lot of work showing that individuals that are minorities experience unique stressors that increase their risk of mental health outcomes. And there were at that time discussions that physiological stress responses would be really important to look at. Up until that point, just 10 years ago, most of the research that looked at cortisol, for instance, was done in the context of HIV/AIDS research. So I asked my mentor, Sonia Dupien, if we could do a study looking at cortisol reactivity and allostatic load among LGB Montrealers. And she said that after 2 years, it took a little while to convince, but she gave me permission. And I was very happy to do this study that really spearheaded a lot of my current research questions in my lab. So I had the chance of recruiting 87 Montrealers, half of which were sexual minorities, half of which were heterosexual. They came into the lab for 2 visits, very similar to the protocol I showed you before. So they came in for a first visit for a blood draw and then went home to do some sampling. And they came back for a second visit where we exposed them to the TRIAR social stress test. And we measured sex hormones 15 minutes before and 15 minutes after the stressor. And what we found was that lesbian and bisexual women had higher levels of testosterone than heterosexual women. No differences in estradiol. And then again, progesterone was higher in lesbian and bisexual women. And this was a sort of an interesting finding that, a lot of research in the 80s was trying to identify sex hormones in relation to sexual orientation. And a lot of that literature was kind of debunked because it was very inconsistent, except that there seemed to be higher levels of androgens in about 1/3 of studies looking at sexual minority women. However, one of the main criticisms of this literature was that psychosocial stress was never actually considered. And so in our study, we looked at measures of questionnaires and found that higher levels of distress were actually related to higher testosterone and higher progesterone in these women. And these higher levels of testosterone and progesterone were actually also related to higher levels of cortisol reactivity. Here you see that lesbian and bisexual women show highest levels of cortisol 40 minutes after the stressor as compared to heterosexual women. By contrast, for men, we found no gay and bisexual men basically showed a blunted sort of stress response, while heterosexual men had higher levels of cortisol. And this is sort of the finding we see most often in the literature. This is adjusting for sex hormones and other important covariates. Now, if we look at cardiovascular functioning, everything's flipped around, where we see that gay and bisexual men actually have higher heart rate. So this is really important from an allostatic perspective because these different systems are potentially recalibrating themselves. So where you might see low levels of cortisol, you're actually seeing higher levels of heart rate. And this is a result we want to go forward in investigating in my lab, but with heart rate variability rather than just heart rate by itself. Now for diurnal cortisol, we found no differences based on sexual orientation. However, I had added a variable asking people about their coming out process and whether they had come out completely to family and friends. And what I found was that among the sexual minority people in my sample, those that had completely come out of the closet had lower levels of cortisol than those that had not completely come out of the closet. And this was shown again in terms of mental health. We didn't find any differences really in mental health between our participants based on sexual orientation. This is a sort of a finding that was a bit surprising given a lot of the literature on sexual minority stress, but it was being driven by the fact that those people who'd come out of the closet had lower anxiety, lower depression, and lower burnout symptoms. Now for allostatic load, we had 20 different biomarkers, and we found again that gay and bisexual men had lower levels of allostatic load than heterosexual men. In terms of depression, we'd found the same tendency where gay and bisexual men showed lower depression than heterosexual men. So this was a bit of a surprising finding. It was not related to coming out. It sort of had us scratching our head. It was driven by lower levels of triglycerides, body mass index, and interleukin-6 among gay and bisexual men. There is a literature showing that as a group, gay men tend to spend a little bit more attention to their diet and exercise, and there's a lot more body image concerns that could be lowering levels of metabolic biomarkers, but it's a bit of a mystery. Of course, this study has limitations. It was really the first study in Canada, and Canada is quite liberal. Stephen Colbert kind of laughed about the study and said that the study would have to be done with real human subjects, so not Canadians. So this really was the motivation for my postdoc working at Columbia with Mark Katzenbuehler, where I wanted to see whether the results would pan out in the US. And we know the US has a lot of variation in terms of where you live, in terms of laws that protect sexual or gender minorities. And so I had access with some people from UCLA to the National Health and Nutrition Examination Survey. So this was a sample with about 14,000 people. And again, we found that gay men had lower allostatic load than heterosexual men. But now because we had sufficient power, we could split things a bit more. And we found that bisexual men were the ones showing the highest allostatic load. Once again, this was driven by BMI, triglycerides, and HDL cholesterol, so metabolic biomarkers. No differences in women. And this was surprising because as a group, lesbian women actually had a higher BMI, but they had actually a healthier lipid profile. So this is a sort of a surprising finding that we want to look at further in the NHANES cohort, but by looking at health behaviors. That's one of the critical limitations of this research, is that we're looking at these group differences or subgroup differences, but we're not actually looking at unique psychosocial experiences within those subgroups. So that needs to be the next step for a lot of this work. This is some fresh data with Mark Katzenbuehler, where we were looking at structural stigma. So based on where people lived, there's 9 different laws that we were able to index from 2001 to 2014. And our idea here was that if you're a sexual minority living in a state that has more structural stigma, we hypothesized that you'd have higher allostatic load. And that's exactly what we found for men. So sexual minority men who were living in states with more structural stigma had higher allostatic load, but we did not see that association among sexual minority women. So this is something we're working on right now. This is just a resume of about 43 different studies looking at NHANES, where there have been some sort of differences found between gay, bisexual, and heterosexual groups in terms of different health behaviors or different health outcomes. And red means that it's worse among that group. Green means it's better. So you can see in the gay group for men, there's quite a few outcomes where higher income, higher education, lower BMI. So these things we want to look at further by taking into account variations in health behaviors and different psychosocial experiences. Now, the last part of my presentation is going to focus on gender identity. And this is some work with Zachary Dubois, my dear colleague at University of Oregon. Zach did really one of the first studies, I think, looking at transgender individuals and allostatic load. First, for cortisol, this was a paper we did a couple of years ago. We found that transmasculine men that experienced more stress with regards to transitioning, more stress with regards to coming out, and more stress with regards to public restrooms had higher levels of diurnal cortisol. Now, there wasn't any significant differences for perceived stress. We suspect this might have to do with whether you're looking at stigma specific to the transgender experience versus questionnaires that are just developed for the general population. So a lot of the work that we're doing together now is trying to really identify unique experiences of stigma to the trans community that are maximally correlated with cortisol levels. This was work done that was in 2 pretty liberal states, so Vermont and Massachusetts. And what we found was that by looking at different biomarkers, this is actually a study that was recently published in Psychoneuroendocrinology, we found that those transmasculine individuals that were living in less progressive parts of those progressive states actually had higher allostatic load, and those that had lower social economic status had higher allostatic load. And so we did this by indexing a lot of information from qualitative interviews in a way that was inspired by some systems models. I just want to point out that there's some really beautiful work being done again by Zachary and Jay Puckett at University of Michigan, as well as some collaborators, looking at individuals in the US before and after the last election, and looking at allostatic load and psychosocial health over the course of 2 years. This is a study that's just starting to come out with some results. And another study is by my colleague, Tanya Podiat, which is an NIH R01 that's looking at transgender women who are HIV positive and trying to understand intersectional experiences of stress in relation to allostatic load. Some of the work that my lab is currently focusing on is stress reactivity among the transgender community. We have a protocol that we're working on right now. It's very similar to the one I mentioned before, except now we want to incorporate measurements of high frequency cardiovascular functioning in order to get a better understanding of cardiovascular and psychophysiology. We also have a study where we're looking at the gut microbiota among transitioning transgender people. And the thinking here is that the gut microbiota is very sensitive to sex hormones as well as stress hormones. And so we want to be able to understand how these changes over the course of transition are related to experiences of gender affirmation. So people are in general going to be feeling a lot better in affirming their gender. And we want to see how that marks out with cortisol, sex hormones, as well as mental health and the gut microbiota. We also have a study looking at sexually polymorphic cognition. So these are tasks that differ between men and women. However, birth assigned sex as a variable is just not enough. We know that these tasks are influenced by sex hormones. We know these tasks are influenced by how masculine or feminine you are. Things get inversed sometimes for sexual minorities. So verbal abilities that sometimes in general are abilities that women are better at. Gay men tend to be better sometimes than heterosexual men. Mental rotation ability is something that men are generally better at, but lesbian and bisexual women actually show strong performance as well. So it really emphasizes that sex and gender need to be considered when looking at cognition as well. Before doing all this work, we wanted to make sure that it was culturally appropriate for the trans community. So we did qualitative interviews with the community, on the one hand to ask them about their health and wellness needs, but also to present them different approaches and different methodologies used in our health research. It was a really wonderful experience. It was my first qualitative study. So we're working on some papers right now that are really going to inform some of the methods that we're going to use. In sort of summary, the one approach that seemed to be the most harmful to the trans community was looking at body mass index. I personally thought that the gut microbiota or high frequency cardiovascular parameters with all the hookups would be potentially more problematic, but the community were actually quite interested in these technologies. BMI, however, reminded them of their birth assigned sex, and that was something that consistently came out in our interviews as something very problematic so we'll simply not include it in our studies. And moving forward, we have my lab has been very fortunate to get some support from IGH for a sex and gender science chair that we're launching in 2 weeks, where we're going to be looking at allostatic load, really with a real perspective of subgroup differences. And we've worked hard over the course of the pandemic to connect with the transgender community in Montreal. We want to really represent as many subgroups within the LGBTQ umbrella as possible, and really try to understand their unique experiences that can affect their stress physiology and their mental health. And the hope with all of this work is that we can move beyond a lot of the sexual minority and gender minority stress models to incorporate physiology, not as a replacement for psychological or sociocultural variables, but as a complement, because we're clearly seeing a different picture with a lot of these biomarkers in our work. And moving forward, it's really going to be important to consider resilience. So coping strategies, social supports, and community resources. And I'm going to conclude with just one small example of this. So of course, COVID happened. So a lot of my work with biomarkers had to be put a little bit on hold. So I got obsessive with a survey that now has 10,000 responses. Within the first 4 months of the pandemic, we reached out to about 3,000 Quebecers. And we found that among the LGBTQ plus community, there was greater experience, more or less across the board, less social support, more perceived stress, more depression, more state anxiety, more trait anxiety, and more loneliness. This sort of was panning out pre-pandemic tendencies. But interestingly, we found that social support was a very important buffer, not only for heterosexual and cisgender people, but the LGBTQ plus community was actually 4 times stronger in buffering the effect of stress and depression. So this is a really important finding, emphasizing really the importance of families of choice and social support. And so this is something that we are going to be exploring further in our COVID research, but also in all of our research to try to identify unique experiences of, not only vulnerability, but resilience within the LGBTQ plus community. And moving beyond the LGBTQ plus community, I think the real take-home message of all of this work is that we really have to go beyond sex as a binary variable. It's simply insufficient in terms of being able to explain all of this, the individual differences that we see in stress physiology and mental health. And so, moving forward, I think we need to think of creative ways to measure gender and to think about sex beyond a category. So with that, just want to acknowledge a lot of the people that were involved in this work during my PhD, and to thank my lab and our sources of support. And I'd like to thank you.
Well, thank you so much for that fantastic talk, Rob. My head is spinning with all that information that you gave us. And yes, thank you. I'm seeing some clapping emojis, and I haven't looked to see if anybody has a chat, but I see that Troy has their hand raised. So Troy, did you want to ask the first question?
I have more of a point, maybe a little bit of a question, but your last bit of data about the resiliency of the LGBTQ plus community in terms of the pandemic, I think also has to deal with the fact that most of us have already lived or barely survived a pandemic.
Oh,
Absolutely.
oh, that's so
right. I mean,
Yeah.
I know what it's like to live with the threat of constant death. So, I mean, I think there's a large part of it, especially the older generation, or my generation, if you want to call me old, there's resiliency because we already survived one.
Absolutely. Actually, the name of our COVID study is Coronavirus Crisis Competence. And crisis competence was a really beautiful theory that emerged from qualitative research, especially among older men during the HIV/AIDS crisis. And the idea of crisis competence is a kind of cultural resilience among people that have faced significant adversity. And so the idea is, on the one hand, sexual minority men would have to have processed their sexual orientation, but then also had to deal with their community dying from HIV/AIDS. And the thinking behind that is that the pandemic might not be such a big deal when you've already had to face the death of your friends. And well, thank you for that comment. It's
I mean, that was my experience personally, as well as my cohort of friends in my age group. We were all like, well, we've been through this before,
Yeah.
In a very bad way, reminds me of my 86-year-old dad saying, I'm used to being lonely, which is not the exact same thing, but the older population, they sort of had lived through wars and things like that. But I'm also wondering, Troy, based on your question, what about like the LGBTQ community? I wrote down, it's the social support is obviously shining through, is really, really important. It's almost like you have to find your own family. Right. So maybe that is part of it, right? There's already this sort of social group that people have found, and that social support just, we all needed that during the pandemic and during all those shutdowns. I wonder if that is also part of it. I don't know.
I think
that was really interesting finding.
I think, though, on the one hand, as cool as that finding is, and as positive as it is, I think we shouldn't forget either that the pandemic has really eliminated
disproportionately
a lot of safe spaces for LGBTQ plus people. So think about trans youth that's stuck at home with the family that is not supportive. That also, I think, really needs to be emphasized. And I think resilience as a construct can be a bit problematic because not everyone is going to be resilient. So I think I just want to caveat that.
No. Did you
Of
do
course.
that by age? Like, did you look at that by, because that could get at that kind of question too.
That's a
right?
good question. So we in that analysis that I put up, we did adjust for age, but adjusting for age
Isn't the same as, yeah.
not the same as studying it as a
Because you
model.
might find a big change, might go.
Yeah, that's a really good point that I'll be able to look at with the subsequent data that we've got.
I am, if there's no other questions, because please don't let me, it always be about me. We have a few more minutes, but my question to you is always going to be, not that I think, my head swims every time I see your presentation because you have so much great data and consideration of so many fantastic variables that a number of us wouldn't necessarily consider. I don't have to go through them all, but whether you disclosed or undisclosed in terms of your sexual orientation, I think that's brilliant. Looking at the sex-specific biomarkers and thresholds, but I guess I'm always, and this is going to be my bias shining through. I'm always, although, okay, I'll come back to that. I'm always interested in the fact that it's the men and males that seem to have a higher allostatic load. Whereas if you talk to a number of us women, we feel like we're the ones that have a high allostatic load. So I still wonder, maybe we're looking at the wrong biomarkers, or maybe it's not just sex-specific thresholds and biomarkers, but different ones that are needed. So that would be one question. And the other thing that I thought was really interesting was that I tweeted out that bit of it because you published it anyway, was which was the occupational, I forgot what you call it, like occupational status, and allostatic load went up in the men, but actually went down in women. And I thought that was really interesting. And maybe it has something to do with social support, but I'm wondering about the allostatic load in the beginning, like when we defining that, are we maybe using the not wrong biomarkers, but do we need other ones? And not just for women, but for gender diverse communities as well.
Yeah, that's a great question. My inclination, sort of my gut feeling, is that a lot of the biomarkers that we were looking at were sort of biomarkers that were normally looked at in men. And I think as we move forward and we have greater representation of immune and inflammatory biomarkers, I think the picture is going to be quite different. I think also we need to, it really is a multi-systemic thing.
All right.
So that also is really critical to keep in mind. So just the MacArthur studies, the successful aging, started off with baseline data, then 3 years, 7 years, 12 years. And what they were starting to see with allostatic load was that the sex differences that emerged at different age groups within that were completely different from one point to the other. So there's all of these variations going on, but then there's also time. So I think moving forward, it's going to be really important to get some of those more emergent biomarkers measured, and then to go beyond using clinical reference ranges that were often based on men. Moving forward too with trans and gender diverse people and representation in the allostatic literature, Zachary Dubois has made a really good point. A lot of these standards are based for men and women, but what do you do with a transgender person? Do you use the average of the cutoff for men and women? What are some more culturally appropriate ways of working with that data? I think too, I mentioned that there's sex-specific formulations that can be used for allostatic load, but hopefully we'll get to a point where there'll be enough representation of trans and gender people in this research that we could actually also look by gender identity rather than birth assigned sex.
Yeah, I mean, the other thing that struck me, and I think there's a question in the chat, but the other thing that struck me, I only got 1 minute, so I should be the one, and this is from Thera, when we say higher allostatic load, does that include burnout where the cortisol curve is flattened at the low end? So
that's a great question. And most of the research that I've done myself, I always will look at how does cortisol pan out with allostatic load. And in a pretty consistent way, higher allostatic load actually does seem to be related to lower levels of cortisol, which is what we see in burnout. So I think There are very few studies that actually have longitudinal data that could look at that over time. But I think that's a really interesting sort of finding from a theoretical perspective, because I think what we're actually measuring with allostatic load when we're looking at cross sections is that this is dysregulation that's spanning years and years, and a lot of downstream effects of cortisol. So it's not surprising that cortisol might not necessarily correlate super well with allostatic load, or that it would just be actually flattened out because it's just sort of burnt out.
Yeah, I think that's so interesting. And then the relationship of how that might change based on sexual orientation and gender identity, all of that, I think is really it could flip, right? It could be completely different. So I think the other thing I wrote down in big letters was context matters. So thank you so much. We're out of time. So I want to acknowledge that and just give you another round of applause. Thank you. For those of you, thank you so much for that. That was really comprehensive and will make me think for like a very long time.
Great. Thank you so much for the invitation.
Oh, great. Always great. Always great.
Bye, everyone.
Bye.